Okay, let's start today's class. Yesterday we have completed protozoa. We have completed this general concepts. We have completed this general characters and classification of protozoa and all the remaining things. Everything has been completed thoroughly. Okay, try to revise multiple times. In today's class, we are going to focus on the porifera. How we are going to move to this porifera means we are going to see the general characters and classification of porifera. And after seeing that, we are going to discuss about the canal system. Canal system in porifera, after that skeleton system, after that reproduction in sponges. After finishing all these things, we are going to discuss some of the MCQ questions. Okay? Okay. Porifera, characters, classification, canal system, skeletal system, and reproduction. This first thing is Porifera general characteristics. Zoologist Robert Edmund Grant, he is the one proposed the name Porifera. Okay, he coined the term Porifera in 1936. What is the grade of this Porifera means lowest multicellular animals. We know you have to remember the outline classification of animal kingdom. In that animal kingdom, we have two different category. One is protozoa and the eumetazoa. In that eumetazoa, further three classification have been classified. In that, one of the term is porifera. Okay, parazoa. In that example, the best example of parazoa is porifera. So, as it comes under the category eumetazoa, it is multicellular in nature. So, this porifera is the lowest multicellular animals belonging to the kingdom Animalia. The word porifera denotes the pore bearers or pore bearing species. See, as the name itself suggests, these organisms contains pores throughout its body. That is why it is said to be as the pore bearers or the pore bearing species. Okay, that particular pores, it is also called as the ostia. This is the general term, but the technical term is ostia. And under this phylum Porifera, there are about 50,000 species. And these are said to be as the aquatic invertebrates. We know this. These are all the category comes under the invertebrate, and they were preferred to live in the aquatic region. And in that aquatic, most of the species are marine and some are the freshwater. Mostly it is marine. Only some are freshwater. Their body is asymmetrical and radially symmetrical with a variety of sizes and colors. As their body is asymmetrical means that we cannot divide the body into two equal halves, or it is said to be as the radially symmetrical. Consider this as the organisms. We can cut these organisms at any plane. Whatever the plane we cut the organisms, we can end up with the two equally morphologically similar structures will be produced. That is radially symmetrical. The poriferans have a spongy appearances and therefore called as the sponges. Okay, the poriferans is the best example for poriferans has sponges because they appeared as a spongy. They appear as a spongy appearance. That is why it is called as the sponges. They are sedentary and attached to the substratum. What is meant by that? The poriferans or the sponges can't move from one place to another. They are sedentary throughout their lifestyle. Throughout their life cycle, it can't move. It makes their place in their one single place by attaching with the substratum. Okay, and that substratum may be the rock. The general characteristics of phylum Poriferans. The first important point is the poriferans or the cellular grade of organization, which means what? Only the cells are going to perform many different functions. There is no well developed, there is no a group of T cells forms the tissue-like structure, and that tissue will going to perform any another function. Only this is the poriferans are the cellular grade of organization, and these cells of this poriferans are the loosely organized. Okay, they have no organs in their body. As it is a cellular grades of organization, definitely there should be no organs. They exhibit a functional division of labor. What is meant by functional division of labor means each cell has their own different function. Okay, we're going to see what are the different types of cells and what is their functions. But each cell is destined to do their own function. That is called functional division of labor. They are mostly found in marine water. Only a few found in the fresh water. They are solitary or colonial. They can be live individually or they can live as a group. Okay, and now the body is radially symmetrical or asymmetrical. We know that, and this body will be base alike. See this question, this line can also be asked in a question. Which type of organisms, which type of phylum, which organisms which is placed under the the organisms which placed in which phylum is looks like a vase like, tubular, cushion like, and cylindrical, and they are nothing but the sponges. Okay, and these organisms are found in many colors like bright red, yellow, orange, pink, or violet, or even white and black. The different types of colors can also be exhibited by these type of organisms. Now, the body bears two layers. One is outer pinnacoderm and the inner choanoderm. Between the pinnacoderm and the choanoderm, mesophyll or mesenchyme is present, and this mesenchyme has different types of amoeba-shaped cells. That is called as amoebocytes. See, this is the most important point. Sponges or the poriferans have two different types of layers. What is it? Two different kinds of layers. One is the outer pinnacoderm. This is the outer pinnacoderm and this is the inner choanoderm. And between this outer and inner, between this pinnacoderm and the choanoderm, there has been present as the mesophyll or the mesenchyme. And this mesenchyme is made up of a different cells, which is called as the amoeba-shaped cells. And these amoeba-shaped cells, this is called as the amoebocytes. Okay, and this is the outer pinnacoderm and this is the inner choanoderm and this is the mesophyll or mesenchyme or the middle amoebocytes. Okay, fine. Choanoderm is composed of the choanocytes. As the mesophyll or the mesenchyme is composed of amoebocytes, this inner choanoderm is composed of the choanocytes. And this, what is the function of these choanocytes? Means they have a collar of microvilli around the flagellum. The most important questionable area is which type of cells have the flagella in it? Choanocytic cells, choanoderm, and this is nothing but the inner layer. Inner choanoderm, which is composed of the cells, which is called as the choanocytes, and that choanocytes have the microvilli in it and the microvilli which is present around the flagellum. And because of this flagellum, the water is pulled through the ostia. Ostia is the pore. Okay, the water can be pulled via these pores by beating of the choanocytic flagella. Okay, because of the flagella, the water can be intaked into the body of the organisms through the pores or the ostia. Clear? As I said, see this poriferans have the functional division of labor. That is why it has been say these poriferans have the functional division of labor means. See, this is the inner layer and this inner layer is called choanoderm and the inner layer choanoderm which is composed of a different types of cells that is called as the choanocytes and that choanocytes have microvilli and this microvilli will be present around the flagellum and the flagella because of the flagella which is present only in the choanocytes the water can be intaken into the organisms through the ostia. See, the function of this choanocytic cell is make the water in and outside of the body of the organisms. See, each cell is destined to do different function. That is why these poriferans have the functional division of labor. Now, mesohylar endoskeleton region consists of inorganic spicules or the proteinaceous spongin fibers or both. Spicules are designed according to the number of axes and according to the number of rays. See, when it comes to the skeletal system of the poriferans, see, it is endoskeleton in nature. What is the name of the endoskeleton? Mesohylar endoskeleton. See, this endoskeleton or the skeleton system of the sponges is made up of two things. One is spicules and another one is spongin fibers. The spicules it is inorganic in nature, that is calcium, calcareous spicules, and the spongin fibers is made up of a protein. That is why it is called as a proteinaceous spongin fibers. Whether this endoskeleton is made up of a spicules or it is made up of a spongin fibers, or in some of the organisms it can be made by both inorganic spicules or the proteinaceous spongin fibers, both inorganic and proteinaceous spongin fibers. Clear? Now, this endoskeleton is made up of a spicules and spongin fibers. So you should know that the skeletal system of poriferans contains the spicules and spongin fibers. Spicules is nothing but the calcium. It is made up of calcium. Spongin fibers is nothing but proteins. Now, the spicules is designated according to the number of axes. There are different types of structure seen in this spicules. Based on their number, it has been named. If it has a one single axon, it is said to be as a monaxon. If it has a three axon, it is triaxon. If it has a four axon, then it is called as a tetraxon. And according to the number of rays, it is said to be as a monoactinal, hexactinal, and tetractinal. Let me elaborate when I deal with the skeletal system of the sponges. See how the spicules and spongin fibers have been, and what is the name of the cells which is going to secrete the spicules and the spongin fiber? See, this will be the important questionable area. The scleroblasts secrete the spicules, while the spongin fibers secreted by the spongioblast. This scleroblast secrete the spicules. This spongioblast secrete the spongin fibers. You have to remember this. Okay. Now, this body comprises numerous pores. We know that as the name suggests, poriferans or the sponge of the sponges have many pores throughout their body. To make through via this pores, the water has been taken in and outside of the body of the organism. And the technical term of these pores is called as ostia. Okay, via this ostia, the water can be enter into the body through the canal system into the central body cavity of the spongocyl or atrium, which opens outside through the osculum. Let me explain this point when I show you the diagram. Okay, show now it's see, these are all the pores. These are all the pores. See the here one pores, here one pores. This is the ostia. These are all the pores, which is also called as ostia. Via this pores, the water has been entered. And this is called as the central body cavity, which is also called as the spongocyl. When the water has been entered all these through all these pores, it has been entered through the canal system and it reaches the central body cavity. After this central body cavity, the water has been taken out through the osculum. Osculum is the place or the part of the sponges where the water has been taken out of the body of the organisms. Okay, ostia is the place where the water has been entered. Via the canal system, it reaches the central body cavity, which is also called as the spongocyl. And that after it reaches the central body cavity, the water has to be sent out, and that is taken through the osculum. Clear? And that is what explained in this point. Now, the water circulatory system of sponges, it is called as the canal system or the aquiferous system. See, the water has been taking in inside of the body of the sponges, and the water has been used for all those mechanical functions or the internal body functions. Whatever the function, the water is essentially needed. Now, the water has been sent out via the osculum. Now, this water circulation inside the body of the sponges has been regulated by the canal system, and this canal system it is also called as the aquiferous system. Okay, now this canal system it is not only a water circulatory system but also it helps in the food collection, respiratory gas exchange, and excretion. Okay, and now there are different types of canal system have been seen. For example, first one is cycon type, the second one is ascenoid type, and the third one is leucon type, and the fourth one is rheumatoid type. There are different types of canal system is present. Okay, in all the types of canal system, the water enters the spongocyl. The water reach the spongocyl or the central body cavity through the ostia. Via this ostia, the water has been reaching the central body cavity, and the water has to be exited out, and that is done through the osculum. Okay, just remember the technical term. What is ostia? What is osculum? What is this spongocyl? And this canal system is also called as the aquiferous system. Okay, now the sponges are the filter feeders. It has been filtering the nutrition. What are all the things? There is a filter which is present in the mouth region of the sponges, and that in that it has to filter the feed. Okay, what are all the things necessary? That is the thing taken by the sponges. And the digestion here is intracellular in nature. What is intracellular means? The digestion which has been taking place inside the cell. That is called as the intracellular. And their nutrition is holozoic, animal-like nutrition. The sponges is going to feed the other smaller organisms. That is said to be as the holozoic nutrition. They feed on the organic particles and minute organisms by the canal system. Okay, by the water current through the canal system, and the food are ingested by the choanocytes and passes to the archaeocyte for the final digestion. Why the food are ingested by the choanocytes? What is the important structure which is present in the choanocyte? Just answer me. Just tell me, what is the important structure which is present inside the choanocytes which is responsible for taking the food inside the sponges? There are two different layers. Of course, microvilli. Another structure is very good. Microvilli and another structure is flagella. The flagella which is present in the choanocytes, and the choanocytes which is present in which layer? Inner layer. And that inner layer is called as the choanoderm, and the outer layer is called as the pinacoderm. In this inner layer, choanoderm, it is made up of a cells which is called as the choanocytes, and these choanocytes have microvilli and the flagella. And because of these two structures, the water has been taken inside the body, and while taking the water, some of the food particles are also ingested by the sponges. And after taking the food and by the choanocytes, it has been passed into the archaeocyte. Archaeocyte is the cell which is meant for the digestion, where the final digestion has been taking place, and that is archaeocyte. Okay, fine. Now, the exchange of respiratory gases and nitrogenous waste occurs by the process of diffusion and the osmoregulation. The respiratory gases have been done through only the process of diffusion. There is no specialized structure has been present in the sponges to done the respiratory function. Take for an example humans. Lungs is the specialized respiratory organ present in the human. But in case of poriferans, as it is a lowest multicellular organism, this respiratory gases, this exchange of gases has been occurred just by the process of diffusion. And the nitrogenous waste products occurs by the process of osmoregulation. Okay, you have no well aware about the osmoregulation. Now the sponge cells have remarkable power of regeneration. You have to remember, you have to just revise the general characteristics of invertebrates. You have known the organism, most of the organisms which is placed under the invertebrates, they have the excellent capacity of regeneration. And regeneration is nothing but the natural capacity to regain the lost structures or lost body parts. And this sponge cells also has the capacity to regenerate its lost body parts. Even if a sponges is divided into multiple pieces or the minute pieces, the cell can aggregate to form again a functional sponges. This is called as the regenerative capacity. Okay, here both sexual and asexual reproduction will occur. Not only sexual reproduction will occur, not only the asexual reproduction occur, both asexual and sexual reproduction has been seen in the sponges. In case of asexual reproduction, it has been done by the fragmentation, budding, formation of gemmules, and the reduction bodies. Everything will be discussed in detail when I deal with the reproduction of the sponges. The fertilization is external. See, there are two different types of fertilization. One is internal fertilization and another one is external fertilization. In case of internal fertilization, the fusion of male and female gamete has been taking place within the body of the organism, within the body of the female organism. In case of external fertilization, the fertilization or the fusion of male and female gamete has been taking place outside the body of the organisms. Say, for example, the female will drop its ovum or the female gamete in the water just like that, okay? Randomly, the male will reach out this female and make the fusion, and it results in the zygote. It results in many of the progeny. This has been taking place outside of the body of the sponges. In case of poriferans, the fertilization seen here is external fertilization. Okay, and the development is direct development, and there are two different types when it comes to the development: direct development and the indirect development. With respect to this direct development, directly the gametes, the young ones, will give rise to an adults. But in case of indirect development, the young ones will undergo many of the larval stages, and the last larval stages will give rise to an adult form. See, this is the young one. The straight away, this young one will grow in size and give rise to an adults. But this is called as the direct development. In case of indirect development, the young ones will undergo many of the larval stages. First instar larva, second instar larva, third instar larva, and after that, it gives rise to an adult. Morphological, this is called as the metamorphosis process. Metamorphosis. Different larval stages, and after that, adult will be formed. That is called as indirect development. But with respect to the poriferans, the development seen here is direct development. Okay. No, but there are also some of the larval stages have also been seen here. That is called as the amphiblastula and Regan larva. And this is called as the indirect larva. So the development seen here is direct as well as the indirect development. Okay. In case it is an indirect larva, indirect development, the larval stages here are the amphiblastula larva or the Regan larva. Okay. The cleavage here seen here is holoblastic. A complete cleavage has been seen here. Once the fusion occurs, the zygote will be formed. After the zygote will undergo the process of cleavage, which means what? Like this. The cleavage will be formed like this. And that cleavage will be a holoblastic. Holo mean complete. Complete cleavage will be seen here. This type of developmental process will be exclusively you can study in the developmental biology. Okay, that's all about the general characteristics of phylum Porifera. Any doubts? Any doubts? Okay. Now that's all about the characteristics of phylum Porifera. Now, when it comes to the classification of the phylum Porifera, there are three classes. This is the phylum Porifera, and the class placed under the Porifera is Calcarea, Desmospongia, Hexactinellida. Okay, in Calcarea, you have to go through the examples. Clathyrina, Leucosolenia, Sypha. Okay, these are all the examples. And Ciliana, Spongia, Spongilla, and the Collina. These are also the examples of the Desmospongia. Euplectella and Hyloneema. These are all the examples for the Hexactinellida. You have to remember the examples. At least just familiar with the name. These are all the things which is placed under the which a phylum or at least okay. Leucosalinea, if you didn't read the leucosalinea here, they can place the question in the exam. Leucosalinea is the organism which is placed in a which phylum. Even if you don't know the classes, if you know at least the phylum, you can easily answer the question. Okay, so just read the name and familiar with the name. These are all the sponges. Okay, and what are all the three classes of Porifera? Calcarea, Desmospongia, and Hexactinellidae. So the first class, Calcarea. These type of sponges are the small and they prefer to live in the marine and the shallow. They are not, they prefer to live in the shallow region, only in the surface, not at the deep. Okay, and this body is cylindrical, vase shape, simple structure, and it exhibits a radial symmetry. Now, these type of sponsors, both it has to prefer live as an individual or it has to prefer as a group. Okay, both things are possible. The body organization is ascenoid, cycanoid, and or leuconoid type. Whatever, either there are different types of canal system. As I said before, either of those type can be seen. Whether it is ascenoid can be seen, whether it is cycanoid can be seen, or leuconoid can also seen, but any one of the canal system present in each individual. The skeletal system is composed of the calcareous spicules. As I have mentioned when I explained the general characteristics of Porifera, I have said that skeletal system is made up of either spicules or the spongin fibers. But in some poriferans, both the spicules as well as the spongin fibers are also seen. But when it comes to the sponges which is placed under the calcarea, the skeletal is made up of only the spicules. It is calcareous spicules, and it is why it is said to be as calcareous spicule means it is made up of the calcium carbonate. And the spicules may be monaxon, trioxan, or the tetraxon. Now, the calcarea is also further divided into two orders, homozoa and heterozoa. You should not need to read these and all. Just remember the example as the Sycon, Grantia, and Lycosolani. Now comes the second class, hexactinellides. See, this is the small, but this is moderate. And this includes the glass sponges. The most important questionable area, just take down where the in which class glass sponges have been placed, and that is hexactinellides. They prefer to live in marine, but in case of calcarea, they prefer to live as in a shallow region, but here in a deep sea. Okay, these hexactinellids prefer to live in a deep sea and they can live solitary. They cannot prefer to live. They should not. They don't like to. They don't like to live as a group, as a colony. But instead, they live as an individual. Okay, and they are pale in color and it grows. The size is not necessary. And see, the body is more or less cup shaped, vase shaped, cylindrical in shape, and they are also exhibit the radial symmetry. And now, see, the body wears branched or unbranched canal system. The exoskeleton is made up of six straight silicaceous spicules. See, the spicules is not only made up of calcium, instead it also made up of made by silicon. In hexactinellate, it is exoskeleton and it is made up of six red silicaceous spicules. Now, second, the canal system here is either it is cycanoid shape or the leuconoid shape, not the ascanoid type. Okay, it is cycanoid or the leucon. The canal system is cycon or leucon, not the ascon. Now, the development includes here is it is Trichimella larva. See, if it is undergo a larval stages, just tell me whether it shows the direct development or indirect development. The larval stages exhibited by these type of sponges is Trichimella larva. So we can say this development is direct or indirect development. Anjaneyulu, answer. Yes, indirect development. The young ones not directly gives rise to adult. Instead, it undergoes the larval stages. So, it is said to be as an indirect development. Okay, if the organisms or the young ones straight away gives to an adult means, then it is said to be as the direct larva, direct development. Okay, the example for exactinelids is Euplectella and the Hylonema. Now, Despospongy. The third class is Desmospongia. This is the largest sized. See, Calcarea is small and the Hexactinellida is moderate size and this Desmospongia is large size. They also prefer to live marine, also the freshwater. They are asymmetrical as well as the radial symmetry and the shape of the body is massive, vase-shaped, curve-shaped, and the cushion-shaped. They may either live solitarily or in colony. They can also live as an individual. They can also live as a colony. Here, the canal system seen here is only the leucon type, and the skeleton comprises of the spongin fibers also, silicaceous spicules also, which are monoxen or trioxin. See, the body is made up of either spicules only spicules, or it is made by only the spongin fibers. But see here in Desmospongiae, these type of sponges have both protein as well as the silicon, both spongin fibers as well as the spicules. So the example here is Euspongia and Spongilla. Okay, this is the class 3, Desmospongia. Okay, that's all about the classification of the poriferans. Now we are going to see the canal system of the poriferans. See, the most important structure which is present only in the poriferans, it is nothing but the canal system. Okay, and it is present in the all the sponges. Is a perforation of the body surface by numerous apertures for the ingress and the ingress of water current. This canal system, the main functions of this canal system is the water circulation. What is the blood circulation system present in humans means? Then it is heart. It is pumping the heart throughout the body of the human. Like in the same way, the water has been pumping um, throughout the organism of sponges by the canal system. Okay, fine. Water circulation system. Inside the body, the water current flows through a certain system of spaces collectively forming the canal system. Okay, the water has been taking inside the body of the sponges via different spaces, and all these spaces are collectively called as the canal system. And this canal system possesses different parts. We are going to see that. And now, before that, we are going to see the functions of canal system. The most vital role is the in the physiology of the sponges is placed by the water current on which their life depends. Circulation of water. Now, all the exchanges between the sponge body and the external medium are maintained by only means of this current. Okay, whatever the thing which has been taken into the body of the sponges or it has to expel out from the body of the sponges means everything has been regulated completely by the canal system. Now, the food and oxygen brought into the body and excreta and reproductive bodies are carried out. Not only the water, the canal system is maintained not only the water but also the water intake and the oxygen intake. Everything has been completely regulated by the canal system. Not only food and oxygen, not only the digestive process and the respiration process, but also the excretion and reproductive process. Everything has been maintained only by this canal system. Now, this current is caused by beating of the flagella of the collar cells. See, this is also an important thing. The inner layer is called as the coinodome, and this coinodome is called as the many coinocytic cells, and this coinocytic cells is called as the flagella, and there are also other collar cells. This coinocytic cells, which is also called as the collar cells. Now, this is the functions of the canal system, and see there are different types of the parts of the canal system is there. What are all the parts of the canal system is? First thing, paragastric cavity or the gastric cavity or the spongocoel. See, these are all the things. You have to remember the other names also. Paragastric cavity is also called as gastric cavity and it is also called as the spongocoel. Now the radial canals is present, incurrent canal is present, Prosophiles is there, excurrent canals is there, apophyles is there. These are all the parts of the canal system. Now you should know which part is doing which function. The paragastric cavity, um, this paragastric cavity or the gastric cavity or the spongiosele. See, if the body of the sponges is bisected longitudinally, if we cut the body of the sponges like this, it is observed that outer larger opening called the osculum. See, this is called as the osculum. The water which has been expelled out via this osculum only. This leads to the large central canal. Just see the example. When the water, this is the place where the water has been taken out. This is called as osculum. When we enter in via this osculum, we can see the larger space, larger cavity, and that is called as this paragastric cavity or the gastral cavity or the spongiosal. This is called as the central body cavity when we this osculum will lead to the central body cavity that is called as the spongiosele. Okay. Now the wall of the sponges is lined by an ectodermal flattened cells. As I have mentioned, two different types of cells has been seen in the sponges. One is outer pinnacoderm and the inner coenoderm. The pinnacoderm it is made up of a cell which is called as the pinacocytes, and the inner coenoderm is made up of a cells which is called as the coenocytes. Okay, that is why here the wall is made up of wall of the sponges is lined by the ectodermal, that is outer layer flattened cell. That cell is called as a pinacocytes. Okay, now the osculum is surrounded by a layer of cells which is called as the pinacocytes, that is outer cells, outer layer, which are contractile in nature, which act as a sphincter. See, in that osculum, this osculum is made up of the pinacocytes. This pinacocytes will act as the sphincter. Sphincter, what is the purpose of sphincter means, which is the contractile inform, which makes their way into the body cavity or from the body cavity to the outer side of the sponges. That has been that this pinacocytes made the way of water from the central body cavity to the outer external medium via this osculum. Okay, fine. Now the spongiosele opens into the exterior through the osculum. The water has been entering into the body of the sponges via the ostia, via the pores. Okay, these are the pores which is present in the body. It is called as the ostia. The water which has been enter into the body of the sponges via this ostia, and it reaches the central body cavity which is called as the spongiosele, and it has to enter outside of the body via this osculum. This is the mechanism. Now, radial canal, the body wall lining the spongiosele, which contains many straight out pushed finger-like projections at regular intervals. See, this is the radial canal. This is the radial canal. Here, this is the place. It is said to be as the radial canal. Now, we should know what is the structure. Finger-like projections at regular intervals. See, this projections is called as the radial canals. The wall of the radial canal is lined with the flagellated collar cells or the pinacocytes. See, you should know which place which has which type of cells and what is the function of the cells. When it comes to the ectodermal cells, it is lined with the pinacocytes. Okay, and this osculum, it is surrounded by the pinacocytes. But in case of radial canal, it is lined with the collar cells. As I have mentioned you before, this inner layer is called as the coenoderm, and that inner coenoderm, it is made up of a cell which is called as the coenocytes, and that coenocyte, it is also called as the collar cells. I have mentioned in the general characters of sponges. See, this flagella or the microvilli, which is present in the coinocyte, it is responsible for whatever the water and the food and whatever the external medium it has been pushed, taken into the body of the organism only because of the beating of the flagella. Okay, so the radial canal, it is lined with the flagellated collar cells or the coinocytes. This point is very much important. In case of oscula, the layer is pinacoderm or the pinacocytes. In case of radial canal, it is made up of the collar cells or the choanocytes. Okay, now the outer end of the radial canal is blind, but the inner end is open, which communicates with the spongocyte through the excurrent canal by an aperture called apophyte. See, the outer end of the radial canal is blind, but the inner, the outer, this is the outer layer. The outer layer of the radial canal is blind, but the inner canal, inner end is open to the sponge. This inner end is opening into the spongocyte through the excurrent canal. This is the excurrent canal, the area where it connects the radial canal and the spongocyte, that is called as the excurrent canal. And this radial canal inner end, which is communicating with the spongocyte via the excurrent canal, by there is an aperture which is present in the excurrent canal, that is called as the apophyte. Now, coming to the incurrent canal. Incurrent canal is nothing but this is the incurrent canal. The space between the radial canal, one radial canal and the another radial canal, it is called as the incurrent canal. In between the two successive radial canals, see, this is one of the radial canal and this is one of the radial canal, and between these two a structure called as the incurrent canal is present. Okay, and that incurrent canal, it is also called as the inhalent canal. And this radial canals and the incurrent canal are arranged alternatively, and the later means what, this incurrent canal opens into the exterior through the ostia. See, let me explain. See, what I have said you, what is radial canal? This is a radial canal. This region is called as a radial canal. And what is the incurrent canal? This region is called as the incurrent canal. See, this is the radial canal. This is the incurrent canal. This is the radial canal. This is the incurrent canal. This is the radial canal. This is the incurrent canal. Between the two successive radial canals, the incurrent canal or the inhalent canal is present there. Okay. Now, this incurrent canal opens into the exterior via the ostia. And that point is, this is the incurrent canal. This is the incurrent canal, and these are all the pores. This is the incurrent, these are all the pores. This incurrent canal make their contact with the exterior via these pores or the ostia. Okay, you should know the inner end of the incurrent canal is blind. In case of radial canal, the outer end of the radial canal is blind. In case of inner canal, the inner end of the radial canal, incurrent canal is blind. Which means what? The inner end doesn't make any contact with any of the region. But in case of, that is why it is said to be as blind. In case of outer end of the incurrent canal, it make their way into an external medium via the ostia. Now, this wall of incurrent canal, it is also lined by the pinacocytes. Okay, as it has a contact with an exterior, it is made up of an outer layer which is called as the pinacoderm, and this pinacoderm, it has a cells, different type of cells. That type of cells is called as the pinacocytes. Now, between the incurrent canal and the radiant canal, there is a thickened mesoglia called the gastral cortex. See, between the incurrent canal and the radial canal, this is an incurrent canal. This is an excurrent canal. Between these two, there is an thickened mesoglia, and that is called as the gastral cortex. Now, the ostia, which is situated on the pore membrane, which is the pores are situated on the pore membrane, which are intercellular openings or surrounded by the contractile myocytes. These are all the cells which is called as the contractile myocytes. These myocytic cells are surrounded the ostia. The cells which is surrounding the ostia, the cells which is surrounding the osculum is pinacocytes. The cells which is surrounding the dermal ostia is called the myocytes. Okay, and this myocytes will act as the sphincter by which they regulate the diameter of the openings. how much the pores has to open, how much the pores has to close, into the to which extent the pore has to open. Everything has been maintained by the myocytes. They act as the sphincter. That's all about the incurrent canal. Now comes the prosophiles. See what is meant by prosophile means this is the prosophile. The wall between the incurrent and the radial canal is pierced by a numerous minute pores called the prosophiles. See, this is the incurrent canal. This is the radial canal. This is the incurrent canal. The radial canal and the incurrent canal can be connected by these pores, which is called as the prosophile. Clear? See. In cycon, each prosophile is an intercellular space. This is one cell and this is one cell. Between these two, there is a connection, and that is done by the prosophile. While in leucocylinia, these pores are intracellular. It has been between the cells and it has within the cell. And through these pores, the incurrent canal opens into the radial canal. See, when you heard about the name prosophile, the only one thing you should make a note of it is the incurrent canal has been made in contact with the radial canal, or the radial canal have been made their contact with the incurrent canal via these pores, which is called as the prosophiles. Okay, fine. The prosophiles or the openings of the porocytes generally believed to the modified pinacocytes. These prosophiles is nothing but they are made up of a cells called the porocytes, and that porocytes are nothing but they have been just a modified pinacocytes. Right? Next. The porocytes are the thick-walled cylinder-like structures with a nucleus in the cytoplasm at one side. Okay, so it has a nucleus in it. This it is highly contractile. As it is very highly contractile, it controls the inflow of water. Okay, this is what a prosophile. When it comes to the prosophile, you have to remember the radial canal and the incurrent canal has been connected with each other via this pores called the prosophiles. That's all. And now the excurrent canal. See, this is the radial canal. This is the incurrent canal. This is the prosophile. Now, what is the excurrent canal? The radial canal communicates with the spongocyte. See, this is the radial canal comes into contact with the spongocyte. This is the radial canal. This is the spongocyte. Through this they are connecting with each other through the short wide canals called the excurrent canals. Here the excurrent canal will be present, and the channel present in the excurrent canal is called as the apophile. See, this is the radial canal, this is the spongocyte, and this is the radial canal, this is the spongocyte, and they have been connected through the excurrent canals, and the channel present in the excurrent canal is apophyles. And this excurrent canal is also lined by the pinacocyte. Okay, now apophile. The apophyles is nothing but the excurrent canal opens into the spongocyte via the internal ostia. It is called as the apophyles. Via these pores, the excurrent canal may send the water into the spongocyte. Okay, now the apophile, it is surrounded by the contractile myocytes. These myocytes will act as the sphincter. As I said before, this myocyte is a checkpoint to how much extent the pore has to wide open. Fine. So the only cell which is present in the wait 1 minute. See, radial canal is the only canal which has the polar cell in it. All other canal is lined with the pinacocytes or myocytes or the porocytes. But the only canal which is present in the coinocytes, all the collar cells or the flagellated collar cells, which is present only in the radial canals. Okay, that's all about the parts of the canal system. If you have any doubt, means you can ask me now. Shanti Karanya, try to be regular, okay? If you have any doubts with respect to the parts of canal system, just tell me. Okay, types of canal system. And what are all the different types which has been seen in the poriferan sponges? What types of canal system is ascon type, sycoin type, and the leucon type. And these are all the extensions of leucon type. We will complete up to this canal system. If time permits, we will extend. Types of canal system. The first type is ascon type. This is the most simplest type of the canal system. See, the most simplest type of canal system. The water will enter through the ostia. There is a coinodum, inner layer, the pinnacodum, outer layer, the mesenchyme, middle layer. The middle layer is called as the amoebocytic cells, and the central body cavity called as spongiocele. And after the water has been entered via this ostia and it reaches the spongiocele, it has been excreted out through the osculum. The most simple type of the canal system is the ascon type, and this type of asconoid sponges or leucosolia. Examples are very important. Don't forget to read the example. So the asconoid sponges or the leucosolia and in the olinthes. Okay, and this in the development of all the synconoid sponges, which means what? If the poriferans have, if consider this as a poriferan, this has the sycon type of canal system. Okay, in the sycon, in all the syconoid sponges, the earlier type of canal system is the ascon type. The ascon type is the one going to develop into the sycon type. So whatever the poriferans which has its sycon type of canal system in earlier of its stage, in larval stage of those poriferans, it must be the ascon type. Clear? Its body surface is pierced by the large number of minute openings, which is called as the incurrent pores or the ostia. We know that all the poriferans which have many pores or the ostia, only through this ostia the water has to be enter into the body of the sponges. And these pores are intracellular spores spaces. Intracellular spaces within the tube-like cells, the porocytes, which extends radially into the mesenchyme and open directly into the spongiocele. Now, this spongiocele is the single large spacious central cavity. This is the spongiocele in the sponge body, and it is lined by the flagellated collar cells. See the radial canal and the spongiocele. They are the only thing made by the or lined by the flagellated collar cells or the coinocytes. Now, this spongiocele opens into the outside through a narrow circular opening, the osculum. See, this spongiocele will make their way into the osculum, located at the distal free end and often fringed with a large monax on spicules. Now, the surrounding seawater enters the canal system through the ostia. Via the spore, the water has been entered into the sponges. The flow of sponges is maintained by beating of the flagella of the collar cells. See, wherever the collar cells have been seen in the sponges, the radial canal and the spongiocele. In these two areas, the flagella is present. Because of this beating of this flagella, the flow of water has been correctly maintained by the sponges. And the rate of flow is slow because the large spongiocele contains much water which cannot be pushed out readily through the single osculum. See, for the entire organism, there are many ostia is present. The water has been entered into the body of the sponges via all these pores. So the water intake will be large. But there is only one osculum to send the water out. So the water which is present inside the body of the sponges is taken out to the body is slow than the intake of water. See, the intake of water is high and the exflow of water is low. Okay, because of the presence of many ostia and single osculum. The course taken by the water current is, see, dermal pores. Via this pores, it is called internal ostia, and via this ostia it reaches the spongiocele, central body cavity, and this spongiocele will take the water outside the body of the sponges through the osculum and reach at the exterior. This is the flow of water. This is the simple canal system seen in the poriferans, that is called the Ascon type. This is the Ascon type of canal system. Now coming to the cycon type. As I said before, what are all the sponges which possess the cycon type in their larval stages? Definitely it has ascon type of canal system. That ascon type of canal system is the one develops into the cycon type of canal system. Okay. Now this type of canal system is more complex. And system of pores and canals is characteristics of cycloid sponges. They say example, Syphi and the Ascan type of canal system is there is no particular meaning. See, what is the purpose of the canal system? See, you should know the meaning of the canal system, not the types of the canal system. See, what does the canal system means? The water has to be regulated. See, that is why you have to attend the class at the earlier stage. These are all the functions of canal system. Functions of what is the functions of canal system is water circulation system. Because of the current, water current produced by the collar cells of flagella, the water current is produced. And because of the water current is produced by the flagella, the water circulation has been correctly maintained in the body of the sponges. And that is the main function of the canal system. No, not only the water circulation, whatever the thing which has to be taken from the body to the out, and whatever the important food, oxygen, whatever the thing which has to be taken from the external medium to the body of the sponges, everything has been correctly maintained only by this canal system. See, but there are the different types of canal system. One is Ascon type, and another one is Sycon type, and another one is Leucon type, based on the uh, development of the sponges. In case it is the simple sponges, it has an ascon type, which means what? There is only there is an ostia which has been taken into the body and it reaches the body cavity and it excreted out through the osculum. But in case of cycon type, there are several critical structures is there, but there is no exclusive meaning. Based on the extra structures present in each type of canal system, the name will vary. But the most basic simple type of canal system is Ascon type. Okay. But the meaning of the Ascon type is nothing. There is no particular meaning. You should know the meaning of canal system. Okay. These are the name of type. Fine. Cyclone type is the most complex type. They see the structure. This is the structure of Ascon type, how simple it is, but see the structure of Cyclone type, how complex it is. Right? And the example for the cyclone type is Syphi and the Grantia. See, it has been theoretically derived from the asconoid type by horizontal folding of its glass, of its wall. See, in this point, what we have to understand is this ascon type of canal system is the one. Ascon type of canal system is converting to the by growing some of the structural advantage, it is developing into the cyclonoid pattern. See, the embryonic development of Cypher clearly shows the asconoid pattern, but later the highest development stage of the Cypher, it has the cyclonoid pattern of the canal system. Okay, so you have to remember from the ascon type, cycon type is going to form. Now, the body wall of the cyclonoid sponges includes two types of canals, incurrent and the radial, paralleling and the alternating with each other. Okay. In the cyclonoid sponges, there are two different types of canal system is seen, incurrent canal and the radial canal. What is radial canal? This is the radial canal. This is the radial canal and this is the incurrent canal. How they are look like? They are arranged parallelly. That is what explaining here. Okay, both the types of canals ends blindly in the body wall but are interconnected with the minute pores. See, this incurrent canal and the radial canal as well as the incurrent canal, they are connected with pores. And what is the name of the pores? Lalita, Anjaneyulu answer. What is the name of the pore? See, multiple times I have explained. Via this pores, the water has been entering, but this radial canal and the incurrent canal are connected by this type of pores, and that pores, the name of the pores is called as what? Prosophiles. Okay, prosophiles. See, everything is about pores, but the pores have different names based on their location. When I asked you how the water have been entered into the body of these sponges, means you have to say ostia. Okay, if I say what is the name of the pore which is going to connect the incurrent canal and the radiant canal, means you have to answer prosophiles. Okay, if I ask what is the name of the pore which is going to connect the spongocoel and the excurrent canal, means you have to say apophile, and that apophile is made up of the myocyte cells. See, the names are different based on their location. Okay, now here the radial canal and the incurrent canal are connected by the pores which is called as the prosophile. Yeah, fine. The incurrent pores or the dermal ostia found on the outer surface of the body open into the incurrent canal. See, the water has been entered into the body of the sponges via this ostia, and it also taken into the incurrent canal. And the incurrent canal and the radial canal will be connected by the pores which is called as the prosophile. Clear? And the radial canal and the spongocoel are connected by the excurrent canal. And that excurrent canal has their pores which is called as the apo. You should know what is prosophile and apophile. Now, these canals are non-flagellated. Okay, this incurrent canals is non-flagellated as they lined by pinacocytes. Only if the cells are lined by the collar cells or the flagellated collar cells or the choanocytes, then it is said to be as a flagellated cells. But in case of pinacocytes, there is no flagella present in it, so these type of canals are non-flagellated and lead to the adjacent radial canals through the mining opening called a prosophile. See, these canals, what is this canals? Incurrent canals. This incurrent canals, there is no flagella present here because they are lined by the pinacocytes, not the choanocytes. Now, this incurrent canals is reaching the adjacent radial canals through the minute opening. And what is the name of that opening? That is called as a prosophile. Clear? Because of this prosophile, the incurrent canal and the radial canal are making communicating with each other. Now, it is not clear whether the prosophiles are channels through the porocytes, but it is definite that in the adult there are simple intercellular spaces. See, here the radial canals are flagellated chambers, only they are lined by the choanocytes. See, incurrent canals are non-flagellated. In case of radial canals, it is flagellated. Okay. In case of incurrent canals, there is no flagella present here because they are lined by pinacocytes. But in case of radial canal, it is made up of it has a flagella. It is said to be as a flagellated chambers because it is made up of the choanocytes. Okay, fine. Now, these canals means that this radial canals opens into the central spongocoel by the internal ostia, which is called as the apophile. Here. Now, spongocoel is a narrow, non-flagellated cavity lined by the pinacocytes. It opens into exterior through the excurrent pore via the osculum. This is the spongocoel. After the water has been reaching the spongocoel, it has to send out via the osculum. See, incurrent canal. Okay, wait. This is the dermal pore. This is called as the ostia. Via this ostia, the water has been entering into the sponges and it reaching the excurrent canal. And this excurrent canal and the radial canal are connected or contacting with each other by the prosophyte. Initially, the water which is entering into the through the ostia, it is going to reach the incurrent canal. Now, the water which is present in the incurrent canal has to send to the radial canal. And how is that possible? Why it is not present means there is no need of flagella there. Okay, there is no current need there. There is no water current need in the incurrent canal because already there is a myocyte. There is a myocyte which is present in the dermal pore. There is ostia. Okay, and what is the function of myocyte means? It acts as a sprinter to how much extent the water has been entered, and there itself the current has been generated and it reaching the incurrent canal. Because why, another important point, why there is no coinocyte which is present here? This is an outer layer. As I said before, the see the part which is present in the outer layer, definitely there is the thing which is made up of the cells which is made up of this outer layer is pinacocyte. The outer layer is called as the pinacoderm. The inner layer is called as the coenoderm. And the cells which is made by the pinacoderm is pinacocytes. And the cell which is present in the coenocyte is coenoderm is coenocytic cells. And these incurrent canal which is present in the outer layer. So obviously there is no point there is a coenocyte has to present there, and there is, if there is a coinocytic cells, if there is no coinocytic cells there, no, obviously there is no flagella is there. Only where there is a coinocytic cell, there is a flagella is present. Clear? This incurrent canal is present only in the outer layer. That is why pinacocyte is present there. Now, this incurrent cells will send the water into the radial canal via the pores, via the opening, which is called as the prosopyle. Now, this radial canal will enter into the excurrent canal via an opening called as the apophyte. Now, the water which is present in the excurrent canal will reach in the spongocyte. So, you have to remember this order. Whether they can ask the question as a chronological order. They have given the option as the ostia, incurrent canal, prosopyle, radial canal. Find the correct order, the correct flow of water. They can be asked like that. Okay, so don't take a chance. Just read this. First, ostia and the incurrent canal, the water which is present in the incurrent canal has to send to the radial canal, and that has been possible only with the opening called the prosopyle. Now the water which is present in the radial canal has to send to the external canal via the opening which is called as the apophyte. Now the water which is present in the excurrent canal reaches the spongocyte, and the water which is present in the spongocyte, the excess water has to send out, and that has been possible via the opening called as the osculum. Clear? Next comes the leucon type, the most complex system. It is called as the leucon type. Ascon is the most simple type, and the complex type is the cycon type, and the most complex type is the leucon type. Okay, this is the order. Leucon is the most complex, cycon is the complex, and the asconoid type is the most simple type. And there is a characteristics of leuconoid sponges such as, this is the characteristic features of the leuconoid sponges. Example, spongilia. Examples, very, very important. Now here the radial symmetry is lost and the canal system has become very irregular. Okay. See the structure. As it is very irregular, there is no correct path of the water circulation. So this is said to be more complex. The flagellated chambers are very small, spherical, and that is lined by the coinocytes. As I said before, wherever the coinocyte is present, flagella and microvilli, obviously there is going to be present. All other areas are lined by the pinacocytes. Okay, now this incurrent canals opened into the flagellated chambers through the prosophiles. And what is this flagellated chambers? Can you name them? Just now I have explained. The incurrent canals send their water into one canal, and through that is through the prosophiles. And that is this incurrent canal is, there is no flagella present here, and that canal will present the will add the flagella. And what is the name of the canal? And this incurrent canal will be connected to the canal through the prosophyte. What is the name of this canal? First, Anjaneyulu, answer for this question. Lalita, Shanti, very good. Radial canal. Why you two people are not listening properly? See, incurrent canal is been connected to the radial canal via the prosophiles. We know incurrent canal has no flagella in it, but radial canal has the flagella in it. Okay, good. Next, flagellated chambers in their turn communicate with the excurrent canal through the apophyles. See, as I mentioned you before, this is the simplest diagram. Let us take this. Let me explain the picture once again, right? See, this is the ostia. Ostia is the place where the water has been taken into the body of the sponges. Clear? Now, after the water has been enter, it reaches the, see, this is also an external dermal ostia. Here where the incurrent canal is present. This is incurrent canal. This is the incurrent canal. The place here it is incurrent canal. Now this the water now via this ostia it reaches the incurrent canal, and this incurrent canal sent their water into the radial canal. See, this is the radial canal. This is called as the radial canal. Initially, it is present in the ostia, and the incurrent canal is present here. And via this incurrent canal, the water enters into the radial canal via the opening, which is called as the prosophile. See, this is the opening. This is the radial canal. This is the incurrent canal. The water which is present in the incurrent canal enters into the radial canal, which is via this opening, which is called as a prosophile. See, there is an yellow structure which is present in the radial canal. What is that yellow structure? That is nothing but the prosophile, sorry, flagella. See, there is no flagella which is present, no such structure which is present in the incurrent canal. But in the radial canal, there is a structure called yellow-like structure, that is flagella. So then a water will be from the incurrent canal, it reaches the radial canal via the opening called prosophile. Now, the water which is present in the radial canal has to enter into the exterior canal. Exterior canal is excurrent canal. This excurrent canal, which is present here via the opening called the apophile. So you have to understand the opening which is connecting the incurrent canal and the radial canal is prosophile. The opening which is connecting the radial canal and the excurrent canal is apophile. And the water which is connecting the water which is sending the radial canal to the spongiocele is excurrent canal. Okay, the opening which is present in the excurrent canal is nothing but an apophile. Now the water which is reaching the spongiocele entering sent out to the body of the sponges via the osculum. Hope you are clear. Now, the flagellated chambers. Here, the flagellated chambers refers to what? Radial canal. The radial canal communicate with the excurrent canal through the opening called the apophile. Okay. Now, this excurrent canal developed as a result of shrinkage and the division of spongiocele which has been disappeared. Okay. Now, this excurrent canal communicate with the outside through the osculum. Though the Leucon type of canal system appears to be modification of the Sycon type. Everything, see, Ascon type will be developed into the Sycon type. Sycon type will be developed into the Leucon type. Clear? See, the modification of Sycon type is called as the Leucon type. And this Sycon type, it is Leucon type, it is calcareous sponges. And the Leucon type is developed directly without passing through the Ascon type and the Sycon type. What we have read, from the this ascon type is developed into the ascon type is developed into the sycon type. Of course, leucon type is the structural modification of the sycon type. Leucon type is also the structural modification of the sycon type. But this leucon type will not develop after the ascon type, after the sycon type. Straight away, the leucon type will be developed. But what we have read in the sycon type, this ascon type will be seen in the embryonic stages of the poriferans, and the after in the adult stage it has been developed into the zygon type. But in the leucon type, like spongilia, directly the leucon type will be developed. Okay, but this leucon type is the structural modification of the zygon type. Clear? That's all about this point. In desmospongia, the leuconoid condition is derived from the larval stages called the rhagon larva. This desmospongia is one of the class of the Porifera. In that, this leuconoid condition is there. In the leuconoid condition, the larval stages which is present is called as the rhagon larva. So you have to understand the larval stages. Larval stages plays a crucial role when it comes to the question in the exam. Rhagon larva is the larval stage of which type of canal system? So then obviously it is a leucon type of the canal system. Now the spongocele of Rhagon is surrounded by the flagellated chambers opened through very wide apophytes. A single osculum open at the top of the spongocele. There are many ostia present in the sponges, but only one osculum is present. Canal system of Rhagon larva does not occur in any of the adult sponge. See, this is an important point. Canal system of Rhagon larva does not occur in any adult sponge. In Desmospongia, leucon type of canal system, it is also termed as the Rhagon type. So the Rhagon type of canal system is also called as the leucon type of canal system. Because it's why it is said to be as the Rhagon type or the leucon type is the leucon type undergoes the larval stage which is called as the Rhagon larva. That is why it is also called as the Rhagon larva. Okay, now comes to the flow chart. This is an important area. See, the water now has been entered through the spores. Now it is reaching the incurrent canal. Now the incurrent canal, the incurrent canal, the water which is present into the incurrent canal has to reach the radial canal, which is also called as the flagellated chambers because it is made up of coinocytic cells. This is the radial canal. The opening which is present, see, the incurrent canal and the radial canal are connected with each other by the prosophiles. We have studied only up to this, but what is prosodus? Only when prosodus is present, these prosophiles can connect this incurrent canal with the radial canal or the flagellated chambers. Clear? Now, this radial canal or the flagellated chambers, the water which is present in this radial canal has to reach the excurrent canal via the opening called the apophyles only when the apodus is present. Okay, only when the apodus is present, these apophyles can make their way from the radial canal to the excurrent canal. Now, the excurrent canal will reach the spongocele and via the osculum, the water has reached the exterior. Okay, just remember what is prosodus and the apodus. Only when this prosodus and the apodus is present, the prosophile and the apophile is become active. Just remember like that. Now, leucon type of canal system presented the three successive grades in an evolutionary pattern. What are all the three different types of leucon type means? Euryphilus type, apodal type, and dipodal type. Euryphilus type means this is the simplest and most primitive leucon type of canal system. In this type, the flagellated chambers communicate directly by the broad apertures, apophyles, excurrent canal. Euryphilus is nothing but the prosophiles. Sorry, prosodus. Okay. See, this is the simplest and the most primitive. In this type, the flagellated chambers communicate directly by the broad apertures apophyles with the excurrent canal. Sorry, this. See, the flagellated chambers. Flagellated chambers means this radial canal. This flagellated chambers directly communicated with the apophyles. Directly communicated with the apophyles with the excurrent canals. No apodus is present here. That is said to be as the euryphilus type. And what is the apodal type means? In this type, the apophile is drawn out as a narrow canal, which is called as the apodus. This connects the flagellated chamber with the excurrent canal. Now, if the apodus is present and they are going to connect the radial canal and the excurrent canals, and that type is called as the apodal type when the apodus is present. The diplodal type means besides the apodus, prosodus is also present. If both apodus and the prosodus is present, then it is said to be as the diplodal type. And it has been seen in the Spongilia ascarella. Okay, that's all about the canon system. If you have any doubt means you can ask me now. Any doubts? If you have any doubts means ask me now. Any doubts? Just say me yes or no. Okay, fine. Uh, that's all about today's class. We'll wind up here. Tomorrow, same time. Okay, we'll meet you tomorrow. Just revise the PPT. Okay, I'll upload in the group. You can refer to that. Okay, thank you. You can leave the class now.